that. So it's my pleasure to introduce Lucia. She studied psychology at the University Catholic in Chile. Then she got a PhD in neuroscience at the Max Planck in Germany. Then she joined Columbia University, and she is now a professor at the Max Planck Institute for aesthetics. Thank you. The stage
Sorry,
is
guys, for all of the technical, problems and etc., and also for not being there. And can you show my presentation or put it on stage, because I wanna say something about you guys. So I wanna first of all start by thanking Anne-Sophie, Clement, Maximilian, and Adrian for putting this together. This is a fantastic conference, and as you have seen, they have done an amazing work getting over all of the weather hurdles yesterday, now of the technical difficulties that we had. And I'm really sorry for not being, where you guys are, hopefully with the better weather in the days that are coming up. And I have particularly good, I'm particularly sad for not being there, because I happen to have been, in fact, in the CuttingEEG in Berlin as a young mother of a 2-month-old baby, and I had a fantastic experience. It was a very difficult moment, to give a talk after 2 months of delivering your baby, but it was great. And also the same baby is the one that today make me actually not come to the conference, because now he's 6, and as most of you know, he is not vaccinated. So I decided that, with my good conscience, even though I love traveling and I love seeing my colleagues and I love seeing you guys and my friends and I love the conference, that I just couldn't afford, the thought that he could just get sick. So I'm gonna do it from here, and I'm really sorry that I'm not there. But I hope that, nonetheless, you can understand that sometimes we are faced with difficult decisions. So. But nonetheless, I want to thank you guys for inviting me and for going through the technical hurdles with me today, and I hope that from now on it runs a little bit smoother. So what am I going to talk about today is what I usually try to say is that I feel that my research is trying to understand why I can enjoy how Michael Jackson dance. And I want you to think about this problem from the idea that when we see an image, or when we're confronted with dance, or with with speech, what actually happens is that we're confronted with continuous streams of information, and then we are posed with many different challenges that we need to that the visual system or the auditory system actually have to solve, which is find the meaningful units for our mind. And the research that today I'm going to be showing, that even though I am here, I am not the one who actually did this. This is the people that I put there are actually the real, heroes for what I'm going to be showing. So what am I interested in? I'm interested in the idea of sequences. I told you in this idea that, how can we understand, how Michael Jackson is dancing? And sequences are ubiquitous, not only for us, as I just told you before, for dance, but also for music, when we speak, when we type or do any movement, but also even when we are engaged in spatial navigation. For all of those, we need to somehow parse the information, organize order, and segment in meaningful units. And yet, it's not really. It seems, because we are trained and we are, and we had, many years of experience with the world, we think that now, because we perceive the world from this adult brain, that this is a very simple problem. But in fact, it's not, if one looks at actually what the world delivers to us. And what the world delivers to us is, like wake forms such as the one that I'm showing you now. There are no silences between words. And I put it there so then you can appreciate that, in fact, there are no silences between words. And if there actually are gaps, very often they do not necessarily reflect the units that then our minds put together. So, for instance, the word silences, as you can see, it's parsed in a completely weird way, or between words, it's also completely parsed in a really weird way. So once upon a time, when we were babies and we came to this world, we were confronted with this problem. So how do we go from this continuous stream of buzzing, sounds, but also buzzing visual images into something that is meaningful for us? And this poses the question of, as I said, like how does the brain parse or segments these continuous signals and creates these units, not only for perception but also for memory? And I don't know depending on how fast I speak today and how much you can, bear with me, I perhaps will be able to tell you about perception and memory, and if not, at least about perception. Then the next challenge is, so once we have found out, say, like the right scissors to cut the information in the proper ways, then we need to integrate information within those segments and segregate information across those segments. And that's yet another challenge that we are faced with. The next one is perception, and language, as you already heard, is hierarchical. So we go, let's say from phonemes, to words, to sentences, to paragraphs, and etc. And all of those different, hierarchical units are in our head and our mind as we understand and perceive the world. And the question is, how are all of those different units processed simultaneously, and at the same time, how are they integrated one to another? And the last one is, how do we organize temporal order? And this is an important thing, for, again, like language, but also for perception. So if you, and you would appreciate, I think in Ed's talk, if you then reverse speech, which in essence means you completely messed up, the temporal order of things, then you don't understand. So it's not the same thing saying things, forward and saying things backwards, or remembering a, birthday party from how it started to how it ended, and then go backwards. And so in essence, every time we perceive and we remember, we need to understand what happened first, what happened second, what happened third, and keep track of the order. And for all of that, we need to somehow understand the mechanism by which that happens. So this is more or less for today. I will try to cover at least some of those, topics. And where did we start? As I told you, we start with this basic idea that when we, now again, we are trained, we are adults, we were really in tune to a particular language. So we learned the phonemes, we learned the syntax, and so on and so forth. But again, once upon a time, we didn't know any of that. And we were only confronted with this, just continuous stream of information. And then eventually we learned that there are syllables. We learned that those syllables in turn compound words, and that those words in turn compound phrases, and that those phrases in turn compound sentences and paragraphs, and so on. And this is the idea of that there are hierarchies for perception, in this case of linguistic materials. Now, the question is, how are those different units being parsed and understood at the same time? And many years ago, with Naiding and David Poppel back at NYU, we started with what we thought was a really kind of like terrible hypothesis, which is that perhaps there is windows of integration, or integration towards a bound, if you want to think about it, that somehow integrate towards the end of a given unit. So, for instance, if you're thinking of a syllable, it will be a very short time window. But if you think about a word, because you have to integrate across two elements, it will be a larger window, and therefore, and the same thing for noun phrases. And if this hypothesis were to be true, we saw that in the EEG, this will be reflected in oscillations that are actually slower and slower and slower, right? And at the time, there was evidence for speech tracking of the envelope in the theta hertz, but we were looking at far slower oscillations in around the delta. It was a really funny experiment. So he brought Chinese speakers to the lab, in this case to an image setup, and he asked them to just sit there and listen to speech materials. But cleverly enough, what he did was to do you see my mouse? No? Yeah. Okay. Great. So what he did very cleverly is that he created the materials in an artificial way such that each syllable would last 250 milliseconds. And so on two of those, which in turn in Chinese were one word, would turn into one noun phrase, and then another two syllables that in turn would turn into one verb phrase. And in turn, the noun phrases and the verb phrases together will turn into a sentence. And therefore, just by the mere fact of having 4 syllables per second, 2 phrases per second, and 1 sentence per second, we would actually be able to frequently tag what the speakers, what the listeners were understanding in this case, right? And when we did this, we thought, okay, this is never going to work, but let's just give it a try. Perhaps it works. And as I told you, the participants would just be listening to this continuous, in this case, like 12 sentences in a row, and would do nothing, just listen, right? And to our surprise, actually positive surprise, we found that it was in fact possible to track the syllables. We knew that we would, because there is energy in the envelope, that there is a strong oscillation at the 4 hertz, but then we also found that there was a strong response frequency in this case at the phrasal rate and at the sentential rate. And this is just not, you could say, like, is this in the acoustics of the materials that we presented? And is not something that, in turn, which what we wanted to assume, which is the speakers who are listening to this, and they are putting these words together, that actually they're creating these units in their head. And so we could somehow, test that hypothesis by looking at what happens when we present the very same materials to people who do not understand the language. So therefore they only have access to the syllables, so to speak, to the acoustics, but they don't have access to the meaning of what they are putting together. And in this case, we did not observe any peak at the 2 hertz frequencies, nor at the 1 hertz frequency, which corresponds to the sentences and the phrases that listeners might have been putting together in their head. And then, when I did this, he did it in Chinese for, reasons that we can discuss later on. But then he also wanted to check, does this work for other languages? Because if this is a property, it should actually appear not just for one language, but for any other language. So he repeated it in English, and in fact, it works just as well. So you actually have here a response at the syllabic rate, and at the phrasal rate, and at the sentential rate. And, in a sense, it works for Chinese, for English, and now this has been repeated, in many different languages, and it works for, Hebrew, for German, and etc. So it's a very robust effect. Now, just because I don't want you to just trust me, but we also looked at the power in the envelope, and what you can see is that, of course, there is a very strong response at 4 hertz corresponding to the syllables, but that there is no response. There is no amplitude increase at the phrasal rate or at the sentential rate in the acoustics of the stimuli. And this is both true for the Chinese materials that we presented, as well as for the English materials. All right. We wanted, as I told you before, we had done this, excuse me, in the EEG, and we wanted to see how much does this translate to a cheaper technique, in this case, EEG. So we used the very same materials that we had used in the EEG experiment, now in 20 more subjects, now with EEG, in this case, like a geodesic EGI EEG, and we observe in essence the same. So if the participants understood the language, they would actually, we would see a very strong response at the syllabic rate, at the sentential rate, and at the phrasal rate, and at the sentential rate, in the case when the stimuli were comprehensible, and in the control condition, then they wouldn't see anything, right? And this works both for power as well as for interval phrase coherence. All right. Now you could argue, well, which is true, the materials that we presented are really weird. It's not that we speak at a given like ta 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 ta, we put intonation, we elongate the phrases, we elongate the words, so we don't have a specific speech rate at which any of these units are delivered. So if this idea of there's a window of integration towards a bound is true, it should somehow generalize to any size in the language, right? And so what Nye did in this case is, again, like very clever. So he presented sentences that would either have 4 words or 5 words or 6 words or 7 words or 8 words. And the question was, would we be able to see these windows of integration that scales up with the length of the sentence, which is exactly what you see here. So now in this case, this is not in the frequency domain, but it's in the time domain. This is global field power. And what you see is that there is what we call like an integration, so to speak, towards a bound that integrates up to the point at which the sentence ends. So, and this is for sentences with 4 elements, 5, 6, 7, and 8. And now if you do the average the other way around, so now if you actually average towards the point at which the end of the sentence is, and you look what happens backwards, it's like as if we see a diminishing in the global field power, which could be understood as, like the integration diminishes the amplitude. But the point I'm trying to make here is that this is more or less a temporal response function that we observe as the integration of a given unit. And this is very nice because, as I told you before, what we were trying to look at is can we get through this mechanism, or through this not through this mechanism, but through this analysis, hierarchical, the integration of hierarchical units? And if this is true, there should be internal organization that is, that goes from smaller units to larger units. And to get at that question, what Knight did in this case was creating, again, sentences that would have 7 elements, but he changed the internal structure. So either he would present noun phrases that would have 3 syllables, or noun phrases that would have 4 syllables. And as you can see here, the integration window lengthens if there are 4 syllables as opposed to 3. But then at the end of the sentence is the same amplitude. So in essence, this is how you can see why we would get the frequency peaks at, say, like the 1 hertz, the 2 hertz, and the 4 hertz, right? Because we can capture these changes in phase and in amplitude. All right, so this is all of these studies we did, so far with MEG and with EEG. But then we wanted to investigate what are the sources of these effects. And for that, instead of looking at a source localization through any of those methods, we thought perhaps, and partly because we actually had access also to the patients, was to present to patients with epilepsy that actually had electrodes implanted in their brain the same stimulus materials. In this case, these are what you can see here is like the electrode reconstructions for each of the patients, and the color codes the electrodes that were implanted on a given patient. And you can see, like how well with these are patients that are tested in NYU, how well the brain is covered in that center, and how lucky we are to then be able to use those. Use this technique to investigate what the sources are. And then again, the patients would just sit down, would listen to these sentences, and would do nothing, just listen to language, right? And now in this case, what we could do, however, was to look at what is the responses of each and every electrode. And you can think again, this is a kind of like what they call like a frequency response function. And we observed 3 different kind of responses. So some of the electrodes would have a very strong response to the syllable, almost nothing to the phrases, and very strong responses to the sentences. Some others, in turn, would have a very strong response to the syllables and to the phrases, but not to the sentences, right? And yet a third type of electrodes would have no response to the acoustics, so to speak, in this case, the syllables, but only would integrate larger units. So in this case, it would respond to the sentences and to the phrases. And I want you to appreciate the diversity of the responses that we see. So some of them respond to acoustics, or very much bottom-up sensory input, but also the others respond exclusively to higher order units that the individuals put in their head, so to speak. Where do these electrodes, so I show you first like the 3 kind of electrode responses that we have, and now you may ask yourself, where are those located in the brain? And this is now for each of those electrodes that we observe with these different patterns, we now projected them onto the brain, and you can see that, of course, the typical candidates that, the language network somehow lights up, the posterior STG and IFG. What is interesting, however, is that we did not, which one could have predicted, we did not observe differentiation in terms of the localizations of these different responses, but they actually tend to co-localize. So for a given STG electrode, you would find one that would do, say, like sentences and syllable, and another that would do phrase and syllable, and yet another that would do sentence and phrases, but not the syllable, right? So the data, they tend to, in our hands, at least it seems as if they are actually intermingled. It doesn't mean it's the same electrode, but it seems that an area it has much more diversity in terms of the responses. Okay, so one aspect that for those of you who do human neuroscience you may appreciate is that very often when we look at our effects, besides what Ed showed, with his very sensitive technique, is that very often we observe effects at the level of the population. But then when we ask, like, can I observe this in every single subject, and is this reliable over time, it's not always the case. What is really cool is that with this technique, it seems to actually be very reliable at the single subject level. So what I'm showing you here is each of these traces correspond to a given individual that we tested, and in all of them we observe very strong responses to the syllable, the phrases, and the sentences, and none responds to when they actually don't understand the language, right? Now, because these techniques seem to actually be fairly reliable at the single subject level, we took one step further, and we thought, can we now use this technique to try and understand how much linguistic comprehension a patient that has a disorder of consciousness still maintains? And this is a very important question, because these are patients that are the patients that we tested. This is Rodica, beautiful studies with them in Cruz. These are patients that have a traumatic brain injury, are 5 days, they are completely unresponsive, but they are non-sedated. And for all of the clinical assessments, they appear as unresponsive. So it's very hard to understand what level of awareness they have, but also what level of comprehension, how much are they oriented toward the external world. So we thought in using this technique to investigate how much these patients may be understanding 5 days after their injury, and also how much this could be prognostic in terms of how well they would recover 6 months after the injury. And so what Rodica did is, in the unit, in the ICU, he put electrodes, she collected EEG and presented this, again, continuous streams of language. It's artificial. So it's made such as, 4 words make sentences, and then, to make a noun phrase and so on. And then she looked at the peaks in the response for the syllables, the phrases, and the sentences, right? And now the question was, could we predict, based on the amplitude responses, on the phrasal, on the amplitude for the phrasal response, as well as the sentential response, which we take as evidence of them understanding something, or at least being able to have sensitivity towards a high order language comprehension, could we use that to predict whether or not the patients will be recovering 6 months after? So she did this, as I said, 5 days into their injury, and then she called them up, or their families, or their relatives, or their guardians, 6 months later, and she collected the so-called GO scale. So how aware, so to speak, they are based on clinical standards. And what she observes, there's a beautiful correlation in which the patients who actually have a better recovery actually had, at the time of injury, so 6 months before, these 20 minutes, cheap EEG, a complete technique, whether or not this would be later on would recover. And this was above and beyond the clinical, the predictors based on clinical observations, as well as their MRIs. So we are exceeding what the medical teams have now at hand, which is MRIs and with this simple 20 minutes. It's a small cohort. I'm not saying that this is how one should end, but I'm excited. I think that this is good news. If this works, with larger cohort, we could be in business in terms of understanding better what kind of linguistic capabilities patients might preserve, even though they are not moving or they cannot respond. Anyway, so with this, I hope that I've shown you some evidence that we, through investigating images and what we call a new kind of high order unit, can investigate. And I told you, for German and for other languages, it's robust. And these responses that we observe, this is not like SSVPs, which actually are the images are changing. This is what the subjects are putting in their head, right? So this is how the participants construct the units, and because we're looking at higher orders of the patients.
Ouch. Lucia, we lost you. I don't know if you still hear me. Lucia! Tadam. On peut la rappeler? Appeler au téléphone? We'll try to make a phone call. There she is.
I don't know why.
Lucia, we see
I
you
was
now.
just kicked out. Cool. Let me just, okay.
Yeah.
Can you?
It's good. Can you put
Cool.
your presentation
Okay.
up?
I hope
Yeah,
that
this
you
is
can,
great.
it goes smoother
Fine.
now. Okay. So what I was saying is that in the last bit is that we could use this, or we're excited that, in a very small cohort of patients, we could use this for prognosis. Now, the next question. So as I told you, we kind of like cheated in some sense, or it felt to me that we were cheating, because we knew we were testing patients, or in this case, subjects that already knew the language, and we knew that they actually had this, they could comprehend these higher order units for language. But the real challenge is, can we use this technique to track a baby who does not have these linguistic units in their head, but is actually trying to get them through exposure to linguistic materials? So we took the next step, and the next challenge is, can we now use the very same frequency tagging to capture when someone is learning a language, so to speak? And this sounds like and this is a long, as I told you, a long, or people are investigating this very much, because babies, and if you actually even think about, when we read text, if there are no pauses, then it's very difficult to parse the stimulus. And when one long tradition, or fairly successful tradition of research in terms of what is the hypothesis for how babies might start parsing language is the so-called statistical learning. And the idea here is that babies are insane bootstrappers, so they can just listen to this completely buzzing word, and they use something like a word to back, which is what Ed showed you before. So they just learn the statistics of what actually follows the transition probability. So what follows a given syllable, and by virtue of doing that, they understand these words. And this started almost 20 years ago, and even longer, by a similar study from Jenny Safran, where she presented to babies a continuous stream such as this one, but unbeknown to these 8-month-old babies, there were like fictitious words that she had created in the stimulus materials. How did she do that? By always having the syllable to being followed by ki, and the syllable ki being always followed by bu. So transition probability is 1, is deterministic. And then after bu, they could actually hear gi, or go, or ti. Therefore, the transition probabilities are lower, so 1/3. And because of that, then the babies would understand that that's a segmentation cue. They should parse that, right? And after that, there's been like whatever 25 years of very fruitful research, whereby people have not only investigated how the statistical learning mechanisms are used to create words, but also to perceive events. And because again, like remember when we discussed the metaphor of Michael Jackson dancing, like how do we know when he's starting, when he's in the middle, and when he's in the end? So in essence, we also have to parse that continuous visual stream into events for the mind. And people have demonstrated based on artificial materials and also more complex materials that similar mechanisms of statistical learning are at play. So both for words recognition as well as for event recognition. And yet, with this technique that we had this neural frequency tagging, we thought this is a good way to now reassess these previous investigations and look at what could be the neural circuit responsible for that, as well as can we find similar mechanisms across the visual and the auditory modality. And furthermore, can we then look for what is encoded within a given unit that the babies, or in this case, the adults, as we tested, have extracted, right? And so what did we do? How did we do this? We actually took the same stimulus materials as Jenny Safran had used in her seminal studies. So these are 12 syllables that are just randomly presented. And the idea was that because they are random, we would actually have a response at the syllabic frequency. In this case, I think it's 4 hertz. But and this is in the so-called random stream, whereby there is no statistical regularities. And we also expose, in this case, patients to the so-called structure stream, where we have these words that we are presenting. In this case, again, because of the transitional probabilities, the idea would be that if they learn that there is certain statistics in the materials, then we would now see us increasing the response at 1/3 of the deaf oscillation frequency. So in this case, 1.33, right? And this is work that Simon Henning conducted at NYU. And we did this again in patients with epilepsy. These are we tested this on 16 patients. You can see here again, like each of the patients is marked with a different electrode, with a different color of the electrode. And again, I hope that you can appreciate like how nicely we can cover the brain through this technique, and in particular with these 16 patients that we tested. And what we observed, much like we had expected, what we had hoped, is that in the case of the random stream, there's a strong response at the syllable and nothing at the word rate. But in the case of the structure stream, there's a strong response at the syllable rate as well as the word rate. And what I forgot to mention is that this is just 10 minutes. So we are presenting these streams last 2 minutes, and we present them for 5 times. So overall, it's 10 minutes of recordings, and the patient just listen to this. They have to do. They have to. Their task is to actually recognize repetition of a syllable that occurs very seldomly in the stimulus materials. Now we repeated the very same experiment, but now in the visual modality. So now again, we presented the patients with, like in this case, we tried to mimic the auditory response by presenting fractals, which are like meaningless stimulus materials. And then in some of them, we created 8 pairs. So therefore the transition probabilities were altered such that we should actually find if the patients have learned overexposure, the pairs, a response which is, like half of the fractal rate. And this is, and we measured this in 12 patients with epilepsy, and these are the electrode reconstructions for each of them. And again, what we observed was that we would have a very strong fractal response, both in the random as well as in the structure stream, but only a response to the pair in the structure stream. And again, this was an exposure of 2 minutes for each stream, and overall 5 times, so overall 10 minutes. So pretty quick. Now, what was really cool, which again, like this is nothing that you predict, but the data show you, is that we looked at what are the temporal response profiles of like those electrodes. And what we found is that some electrodes, much like in the case of the linguistic materials that I showed you before, would have a strong response to the syllables as well as a strong response to the words, whereas yet another set of electrodes would have almost no response to the syllables, but a very strong response to the words. So in this case, they don't have any bottom-up responses, but they integrate towards the words, right? And what I'm showing you here is the electrodes localization of these 2 types of responses. And now, contrary to the previous example where I show you that they all intermingle, in this case, there's a very strong separation between the word-only responses, which are actually outside STG, and the word plus syllable responses, which are predominantly just in the STG. Right. So in this case, there is a hierarchical separation between these 2. And we observe a very similar, of course, in different areas, but a very similar organization of electrodes responding to the fractals and to the pairs, now in this case, localized in visual cortex, and also on a set of electrodes only responding to the pairs that are actually localized not in visual cortex, but outside, and tend to actually not co-localize with the ones that respond to what we call like high order units and lower order units. So both for the auditory materials and for the visual materials, we observe this hierarchical separation in terms of temporal tuning. Now, again, we did this, with ECOG, but how likely we are to observe this in normal subjects and with other techniques. We repeated the auditory experiment in seven normal neurotypical subjects, doing EMG in this case, and these are the frequency responses that you can observe. And very nicely, we can also recover through intertrial phase coherence the different response profiles. So some components respond to the syllable and to the words, whereas some respond only to the syllable, and yet another one respond only to the words. So very similar temporal profiles also observed during the EMG in neurotypical. So this is, it seems to be generalizable across populations. Now, what about the visual statistical learning? Now, you would argue that perhaps it doesn't because vision is different from audition, perhaps we don't observe the same temporal response profile. That's not the case. We actually, when we repeat the experiment, in this case in 12 subjects with EEG, again neurotypical, we observe the same response profile, the same separation response profile. So some components responding to the fractals, some are responding to the pairs, and some are responding to both. But now you could, one thing that I forgot to mention, but I'm sure that you may have noticed, is that in the case of the visual response, in the visual statistical learning, we only presented pairs. We did not present the triplets, as we had done in the case of the auditory experiment, right? This was for, because I was scared in principle that perhaps the patients, if we present the materials too slow, which would have meant that perhaps we couldn't really see the frequency tagging. That's why we opted for pairs. But still, it begs the question: does it generalize to stimuli that are actually slower, right? And it does. So if now we present, instead of pairs, triplets in the visual domain, now of course we have a slower response, but it's still nonetheless observable. In this case, again, we did it with neurotypical, with EEG, and with 12 subjects. So it seems that it's not bounded to a particular response frequency. So it can be observed at more response frequencies, but this is, yet to be seen. You will see that, that there is a limit. So it's not, so it works for 2, it works for 3, and you will see it doesn't work for 4. and we will see why. Okay. So what is great about this frequency tagging is that it allows us to somehow scout, places in the brain or areas in the brain that are sensitive to these different windows of integration, but it doesn't tell us what has been learned or what is being encoded in these different response windows, right? And this is not a trivial question, because even if you take the simple statistical learning paradigm that I just showed you, what subjects can learn is at least 3 different things, and they are all equally compatible with observing a frequency response at the level of the words. So they could have learned the transitional probabilities, meaning that one syllable actually has a much lower transitional probability as compared to all of the others, and this would be coherent with a response frequency at the 1 hertz. They could also have learned the ordinal position. So knowing that the syllable tu, the syllable bi, go, and pa are always the first position, whereas pi, da, la, and do is always the second, and ro, ku, bu, and ti is always the third. This will also be compatible with observing segmentation of the word rate. Or they could have actually learned the words as such, as a chunk. So tu, pi, ro, and then been a distinctive stimulus as compared to bida, ku, go, la, bu, and pa, do, di. Right? And all of those 3 are equally compatible with what we observe at the neural frequency tagging. So to get at this question, what Simon did was now representational similarity. And so he looked at the responses of each of these syllables, and he was looking at, if you want, what is the confusion matrix that we observe, right? And he looked at, first of all, like the electrodes that would have responses to the syllables and to the words. And what he observed is that in those electrodes we have a very strong confusion, so to speak, based on the first syllable versus all of the others. So the one that actually has very low transitional probability as compared to ones that have a high transitional probability. In contrast, in the electrodes in which there is responses only to the words, you observe that across 2 different dimensions, we could recover either the ordinal position, so this is position number 1, position number 3, or position number 2, or the words themselves. This is 1 word, a 2nd word, a 3rd word, and a 4th word, right? So what is nice is that it seems that this hierarchical separation is not coding for the very same representations. It's representing actually different aspects of the sequences. This leaves open the question, what happens in the visual domain? And we observe, of course, the electrodes are in different places in the brain, but what is interesting is it's the same separation or the same response profile, so to speak. Now, again, the electrodes that respond to the syllable and to the pairs in this case encode only the transitional probability. And in contrast, the ones that would respond only to the pairs would encode in 1 dimension the position. So in this case, it is the first position and the second, and in another dimension, the words themselves, right? Now, we also looked at the hippocampus. We had a hypothesis about why. I can, in the interest of time, I'm not going to go into that, but we had a hypothesis that the hippocampus should actually encode the chunks and only the chunks, and this is exactly what we observed. So this is for the auditory domain. As you can see, this is one word, another, and another, and this is, again, the 4 different pairs that we presented to the patients. So with that, what we observe is that across different areas in the brain, cortex and the hippocampus, there's different ways in which these very same sequences are being represented, either at the level of transitional probabilities or order or chunks. Right? I'm just gonna, just a brief note on, when we do direct cortical stimulation, trying to obliterate the response of the hippocampus, we actually can, diminish significantly the responses in cortex at the word rate. So we think that the hippocampus is really relevant for the acquisition of these regularities. But this is a side note. All right. So with this, I hope that, I was able to show you that, that we can, with this neural frequency tagging, we can not only test once someone has learned a language and what are the capabilities that are remaining either in a non-responsive patients or in us, who have already learned the language, but also how we learn that, right? But at the same time, demonstrate that the frequency tagging is, while it's good as scouting and like allowing us to have like a nice marker, it doesn't necessarily tell us what is within those windows of integration, and to that we need to use other techniques such as representational similarity or such, and that at least in the field of statistical learning, there has been a long debate as to whether people learn chunks or transitional probabilities. At least our data seem to suggest that the brain doesn't choose that. It just learns all of those at the same time in different places. But it actually learns across the hierarchy all of those different sequences. I wanna briefly show you something that a colleague of mine actually did, because they took the frequency tagging, but now instead of actually looking at the EEG, they look at pupil dilation. And now they looked at statistical regularities in the visual domain. They presented pairs of faces, and their hypothesis was, so if I present stimuli that do not have any structure, perhaps the pupil will entrain to every image. But as we are learning the statistical regularities, perhaps the pupil will also entrain to the acquired pairs or to the acquired higher order structure. And if that is the case, then we should actually also see a modulation frequency at the level of the pairs. And this is what you can observe here. So this is just one single subject when you present a random stream. And this is an example pupil diameter for when subject is exposed to a structure stream. As you can see, a very nice, like a temporal organization. And when you then do this into an FFT, then you can observe in monkeys, in a cohort of 30 subjects, strong responses to the images as well as to the pairs. And they also took it, like in monkeys, and they observed the very same responses. So I want to give this as a side note, because I think that this neural frequency tagging is perhaps one interesting technique by which we can also conduct cross-species studies, and rely not only on EEG, but also on other metrics, such as, for instance, pupil dilation. They went even further, and to do something that we hadn't done in our studies, which is much nicer, which is to look at what are the perceptual consequences of, this learning or segmenting the materials, or the distributive learning that occurs, while we're presenting the stimulus. And so to get at that, what they did was presenting a continuous stream, and they just ask subjects to detect a given image. But unbeknown to those subjects, there could have been 3 different conditions. So either the trained pair, a foil, meaning that there's the same, they are seeing a face that had been shown before, but it was not part of the specific pair that they would have predicted, or a completely novel pair, right? And what they observed is that they're looking at the very same image, right? And the question is like how easily they will detect this image. And what they observe is that in the case of when the subjects are presented with the trained image, they can detect this given image much more, a little bit less when they are presented with a foil, and far worse when they are presented with a novel stimuli, which means that in turn they are using the predictions set up by the previous stimuli to better detect the stimulus that follows up. they did something even nicer. So they looked at the correlation between this effect in terms of, how much the subjects can predict the trained pair, as in correlation with the frequency tagging that they observe at the acquisition time. And there's a very nice correlation between, in this case, this is the size of the pupil dimension or the frequency tagging that they observe, and this is how much the other way around. This is the pupil, and this is the accuracy. So they actually can denote that there's perceptual consequences for this, which we couldn't actually demonstrate in our studies. All right, so, and the last bit that I want to show you, this is a study that, Lucas Benjamin and Ghislaine Dehaene actually had conducted, which is really cool. They used this frequency tagging technique to try and investigate what happens in neonates, right? Because as I told you, like the real challenge is, there is babies out there who cannot understand language, but they should figure it out. So they expose babies to, in this case, statistical regularities again, but as opposed to 3 stimuli, which is, or a word composed of 3 syllables, which is what most people use. They presented the babies, in this case, and adults as well, with words that are composed of 4 stimuli. 4 syllables, right? So they did a longer window of integration in this case. And they had 2 conditions, one in which they presented the stimulus continuously, so there's no pause, there's nothing that denotes the segmentation, and another one in which there is an almost imperceptible 25 millisecond pause at the end of a given word, right? And what they observe in adults is that when there is no pause, the adults can distinguish between a word and a part word, and what they call a completely shuffled word. So there is some indication that they learn, but they haven't really completely understood the structure, because a part word means that they have learned the pairs inside the word, but they cannot still put together the whole chunk, right? Whereas with a pause, the adults could really figure out, in this case, when they are listening to a stimulus, which is a real word. So in this case, like A, B, C, D, as opposed to C, D, A, B, right? Now, what happens to the babies? This is again frequency tagging. This is Empower and PLV. These are 1 to 3 days born neonates, just listening to these materials, in their cribs or either in the lab of the experimenters or of the mothers. They do nothing, they're sleeping, and we're just collecting the EEG. And without a pause, we actually did not observe any signatures of frequency tagging. Whereas with a pause, there is actually very strong evidence for frequency tagging. So in this case, it seems that the babies need somehow, we interpret this as a failure in working memory, that they cannot integrate across such a large unit, but only across smaller pairs, and that's why they fail to, we fail to have a frequency tagging. So. Because of the session that I'm speaking in, I want to somehow, advertise this so-called frequency tagging for higher order mental operations, and I hope that I gave you good, or some evidence that this can be used to, for instance, look at language, look at learning, but also, which I think is the major challenge, is like, how can we try to get at people who may not even understand our instructions, people who are not necessarily cooperative, such as babies, unresponsive patients, but other, populations such as monkeys or mice, because my hope would be to find techniques through which we can do proper comparisons across populations that may not necessarily follow our instructions. So, and with that, I want to wrap up, and I hope that I show you some evidence for, how we segment continuous signals, how these are integrated, using frequency tagging to track higher order perceptual organization, in this case, linguistic organization, but also temporal order. And overall, like what we're trying to do in my lab is to think about the brain as opposed to think about language and, like audition, vision, and attention and those complicated cognitive operations. Think about basic computations that the brain may be performing. And so we're thinking about, what about, tree structure, hierarchical clustering, 1D linear mapping, or logical operations, and see and understand whether or not those functions can be at the serve of many different cognitive functions, such as attention or language or perception of, events and so on. And with that, I want to thank the people who actually did all this, my collaborators. And I just want to, because I have the mic, I want to tell you something about another project that I'm leading, which is called Cogitate, where we're doing an open science adversarial collaboration. And we have 2 open positions, one as a scientific software engineer to help us figuring out metadata and bits and understand, like not only what the data are, but also what the metadata should be and how we should convey those. And so if you're interested, please apply to our position by the 1st of November. And then Stan also has another postdoc position for this adversarial collaboration. And with that, I want to thank you. And thank you for bearing with me, and sorry for all of the technical hurdles.
Okay. We have time for a few questions. No questions, so you want?
We can go to the round table.
Okay, so we can switch directly to the discussion. So I will ask the speakers from the symposium today, this morning, to join us on the stage.
Just as a check. Lucia, can you hear us? Okay, good. So I'll try to, because I have the screen on my back, so I'll try to just wave your hand when you want to, to say something, and probably keep your mic down the rest of the time, so we avoid interference. So the idea is to have 1 hour and a half of debate now, and then go directly to the next session. No, because we had actually a problem with the lunch and the catering, they just got flat tires, so there's no food, and so I'm sorry about that. So, no, it's a joke. So, so a few people were starting to believe, hardly ever serious. So, I don't know, I'll try to I have a few questions, and then let's just see how it evolves. I start with a positive question, because I'm known to be very negative and try to destroy everything and say science doesn't work. But no, it concerns the presentation of Ed and of Stephanie. So I was thinking, and there is a few questions inside it. So you were talking about partialing out the auditory signal, the auditory low-level representation in a way. So you didn't tell us how you do it. That's sort of magic, but that could be sort of part of the answer. And I think a lot of people will be interested in that. But then my question is, would it be possible, because one of the advantage of this technique, which is also the case for Lucia with the tagging, is that we can work on more ecological conditions. Yeah. And now Stephanie here, she's struggling and asking people to repeat the name of a picture in her experiments. And in even this case, with highly controlled things, she has a lot of struggle to get rid of the muscular artifacts. So would it be possible to sort of combine and to consider in a way the muscular, so the output, and to partial out the output, and then maybe in the future to think that this sort of technique could be applied to continuous speech production?
Yeah, this seems to be working. Okay, yeah, so I think definitely we've thought about that a little bit. We haven't made a ton of progress on it yet, but obviously there's challenges. The more correlated in time the signals are, for example, the more difficult it is to partial things apart, because if they're all exactly locked on in the exact same time course, then you're relying on fluctuations in the time course in order to pick up signatures of them in the brain data. And so if they're doing exactly the same thing at every moment in time, it can be very difficult. The bits where we've thought about it in the past, we thought about it in the context of speech production in terms of people generating the semantic ideas that they wanted to express, and could we pick up on those? And I felt kind of a little bit more hopeful, because it shouldn't necessarily be the case that the amount of meaning in every word that you're about to speak should correlate all that strongly with the amount of muscle you're moving in order to say that word. So there'll be some correlation, because every time you say a word, you're moving, but it won't be a perfect correlation between the amount of meaning associated with each word and the amount of muscle movement. And then, now you've got differences in those time varying signals, and those differences now allow you to pick up unique variants in the brain data that you're seeing to each signal, because they're not perfectly correlated with each other. And so, yeah, for partial correlations, we often just, honestly, we just use partial car in MATLAB. It's a function where you can feed in, an X, Y, and a Z, where X is what you're interested in, Y is the other nuisance variables that you want to control for, and Z is the brain data that you're trying to predict, for example. And there's lots of documentation about how the mathematics works underneath that. But yeah, I think you could do it for speech production, but it's definitely challenging. And I mean, one thing that I didn't talk about that Stephanie's obviously been making great progress with is we don't often tend to do all that much pre-processing and looking at actually cleaning the data in advance, which we probably should do more of in general. One of the advantages of the TRF, though, is that, again, even if there's noisy signals in there, if they're not correlated in time with the thing you're interested in, they're just not going to show up in the analysis. Still, there's no good reason not to do them in the first place. Yeah.
If you want. You can give your telephone number to us so you can start a collaboration straight away.
Yeah, no, just for the blinks, for example, this just made me think of the blinks, that people tend to blink when they respond. So that's something that you would have, not just, even if they're not speaking. But yeah, it would be worth it just looking at how correlated are the artifacts compared to the brain signal. Yeah.
I mean, yeah, and you would then tend to see, obviously, a bunch of strong predictors or channel weights, decoding weights up over the front, which we tend not to see. But definitely it's, we've noticed, for example, in some of the attention studies we've done, people will have a tendency to want to look a particular direction. If they're listening over here at the corner of their eye, you can get some funny behaviors on the part of subjects. So we do, we claim our studies are somewhat naturalistic when we're doing that stuff, but definitely we try to control certain things like that could contaminate the signal in a way that makes it hard to disentangle. Yeah.
I mean, by definition, it's hard to control naturalistic, right?
Absolutely.
Can I say something? It's more a question, for you all as, for us all as in the audience as well. Sometimes, I think that part of going naturalistic is understanding that, the brain perhaps does, compute things in a particular way. So perhaps the eye movements that we see are inherently related to how we, parse signals. So this is what I was trying to show, for instance, with the frequency tagging, right? So, and with this, I want to invite us to think about what we consider an artifact. So I guess that's what I'm trying to get us, your ideas on, because perhaps the fact that we blink could even be, in my wild speculation, be that, like you reduce attentional resources around that, and because of that, then you can parse something. I don't know. Like I'm just, somehow like coming up with some random explanations, but how do we understand artifacts? I guess that's what I'm trying to ask.
I mean, I think the limitation is that the blinks or the EMG are so much bigger than your EEG signal of interest. So it would be hard to say anything about what the brain is doing when you have that artifact, because it's just going to take over the power of the signal.
So of course I understand your point in terms of the signal to noise ratio, but what I was trying to make us think about is perhaps if you throw all of those into your classifier, you would actually get information that is relevant as well.
Sure. Yeah. Maybe people blink harder when they're in a more difficult
or as I show you, for instance, with the pupil dilation. So that seemed to, so I don't know how when we think about, about that, is that an artifact? it seems that it's an attentional, or you can think about it as an attentional modulation that then impacts the way in which patients, in this case subjects, segment the visual signals, right?
Yeah, I can think of one example, all right, that I think might speak to that. There was a, yeah, I apologize if this seems a bit aggressive. I saw a paper published recently that claimed to decode attention substantially faster and more accurately than we had ever done it, which is great. I'm delighted to see papers like that coming out for sure. But this paper, it seemed to me, the results were driven by like beta power activity right here and here. And, I was like, what kind of EEG is that that's driving this thing? So I actually inquired about it and discovered there was no control over what the subjects were doing with their eyes during this experiment, and these stimuli were spatially separated. And no, I have no proof other than that, but it was almost impossible to believe how good this decoding was working. And it's out there as a paper, decoding attention massively quickly. I don't think they're decoding attention at all. I think they're decoding what these people are doing with their eyes during this experiment. And sure, if that's what you want to use to decode attention, that's fine. But you can misattribute your signal then as something else by mistake, and write the title of the paper that they've written, which I don't think is correct. But so yeah, it's yeah, I take the point, but I think in interpreting what you're seeing, it definitely pays to have some control and be able to disentangle X from Y to know what's what. I should just clarify as well, these partial correlation things are not magic either, and we're very potentially guilty of misattributing certain signals in our minds too, because we say that we account for the acoustics in the speech and we regress it out of the EEG responses, and now we're left with these linguistic signatures. And I think there's some truth to that. But if we have imperfectly modeled the acoustics and just not appropriately captured all of those acoustic variations, lots of different dimensions acoustically that you can think of in speech, and then we soak up some variance now with our phoneme predictor, we might now think, oh, this is great evidence for categorical phoneme level processing, but it's not absolutely sure that, because you have to account for everything, and it's impossible to account for everything. So yeah, so one needs to be careful with building these models and interpreting them, and always have your discussion paragraph that highlights your limitations and all that stuff, which we do try to do.
I have a more hot topic, maybe mostly for Ed and Lucia. And so how do you see the future of what you presented? So TRF approach, or for Lucia frequency tagging. For me, the seen from outside is there's a sort of a limitation in both approaches, which in a way both is using ERPs. But in more ecological conditions, because also what Lucia, tell me if I got it wrong, but for me, so tagging the syllable level is you have an ERP for every syllable, and then if you have an N400 every 2 syllables, then you're tagging the word level if it is bisyllabic words. And for Ed, so you have this, you're convolving with this temporal response function. But so in a way, we are still in the ERP framework. Which is a certain type of framework, but it's sort of static framework. So for me, there's missing the dynamic in a way. So how the information flow from where then cognition and perception emerge. And so how do we go there? So first of all, tell me if I'm wrong, and do you have ideas on how the community can move further?
Yeah, no, I think certainly I feel that's a very fair interpretation of what I was pitching. The signal is the signal. It's the same in general, to a large extent, at least the same thing that we see in ERPs, and where we try to separate it out within the same experiment into components that reflect, say, acoustic processing versus semantic stuff. People were already doing that by designing experiments where they present words, and sometimes they're incongruent, sometimes they're congruent. They get a difference wave. It's the same basic principle. I do think, and I think there's other cons with our approach where we assume a lot of kind of linearity and things like that that are not likely to be accurate. And then there's pros where we don't have to repeat trials in this way, unnaturalistic way over and over again, and have people making other meta judgments about the congruity or otherwise of the words rather than just the semantic processing of it. So I think they're two sides of the same coin. I mean, going forward, I should say it's not just you know, time resolved low frequency stuff, we will often look at alpha power changes and beta power changes over the eyes and things like this that fluctuate in accordance with the experiment. But again, that's still similar to time-locked EEG work that was looking at intertrial phase coherence or whatever it might have been. So yeah, I don't quite know. I mean, there's definitely more we can do in terms of the modeling piece. You know, these linear models are definitely wrong. You know, we have actually published some papers showing that our envelope model gets better if you incorporate different nonlinearities to it and all that kind of stuff. You know, machine learning obviously has potential, albeit EEG is a low dimensional enough signal and care needs to be taken there. So yeah, no, I think it's a spicy enough question. I think, but I take the point for sure, and I agree. I think the pros are the naturalistic nature of things, hopefully adding interpretability to and disentangling the EEG signals, but it still ends up with, you know, driven by the same scalp-based things we've been looking at for years, because what else?
I mean, you know, my sense is, you know, you're making an extremely good point. You know, I don't think that this is just it's not a fancier thing. It's just, as I see it, at this, for instance, in the case of the neural frequency tagging, is that you can have less trials. It just takes you less time. So I do think that in, you know, from the pragmatical point of view, it has certain benefits. But I was also trying to show in my talk that it's limited, right? So it doesn't tell you what it is inside, much like any ERP. Any N400 tells you like what, you know, what are people decoding there or understanding or, you know, like integrating or whatever, right? So I think that we do need better methods. And on the temporal, you were asking a really good question, which is like, what would be like a better spatial temporal method? And I don't really know. You know, but at least what I'm trying to investigate now more is like what is being represented. You know, I think if I can crack that question first, then I would love to understand in time how is that being composed. You know, and then how do I go from every syllable, and then you know how that changes. You know, when I present like, and then the next phoneme and the transform like, you know, whatever, like one form morpheme into another morpheme. You know, great. But minimally, I would like to understand what is the operation that is being performed. And I think that the representational similarity stuff are actually pretty interesting.
So maybe we hand it to the public, yes, on the nature of representations. And Anka Klen has something to say about what the nature of representation is.
No. Well, thank you very much for this really nice symposium, first of all. I think I have a sort of very general question that I can then transfer to a more specific question if that's too big. I find it's really, really nice to be studying how the brain reacts to continuous signals, because basically that's what we are doing in real life. And my favorite continuous stimulus is not language, it's music. So the general question is, how about music? And maybe I can make a more specific question. In the talks you've been giving, you have these very structured hierarchical models of what language is, and we have phonemes and syllables and blah, blah, blah. We can make sort of the same claims in music, but it's perhaps less of a strong claim, and it might depend on the musical culture and the people listening to music themselves. So my question is, can we hope to gain some insight from the type of techniques you're using on how we chunk information in music? What are the meaningful units for music cognition? Does that make sense?
Yeah, thanks for the question. I think we've thought about it a little bit. We've recorded some data and started to try to make some progress, and other groups are doing similar. And I think, yeah, I think it's very interesting. What are the units of phrasing? I mean, obviously phrases in music, and you have verses and choruses and things like that that have structure that people can recognize. And so we've begun to look for signatures of that. We actually almost fell at the first hurdle by having difficulty in modeling the EEG responses just to the acoustics, because the acoustics can be, you know, we had so much success with this envelope speech thing. You know, but music, often rock music or something, for example, is like a wall of sound. There is no envelope as such across the whole thing. And there's envelopes within the songs, the vocals and so on, but they're still masked by all this background rock music. So even just getting basic markers in the first instance was tricky. I think we've made a bit of progress with that now and parsing things out. But yeah, and so we've been thinking about things like a little bit like our semantic work with speech, where a next word based on its context is surprising or carries a lot of information, semantic information. You can also quantify using algorithms these days how surprising or probabilistic the next chord or note sequence was as a function of the previous context. And so people have started to do that and find some little hints that if you take that into account, you can better model EEG responses to music. And it provides at least a framework now that you can quantify, parameterize in some sense what's the probability of the next phrase or the next note, and go looking for signatures of that. And I think that's a really interesting question, right? Because speech and music are so different in that way. Once I've heard someone talking, I don't really need to hear what they said again, maybe a second time, but certainly not a third or fourth time. I've got the information. I'm not surprised at all anymore. Whereas there's some songs I've listened to like 100 times. And even though I know, strictly speaking, what's coming up, it's still surprising to me physiologically as a reaction. I think that has a lot to do with the kind of history of, that I've grown up with in Western music. And I know what the traditional chord transitions are. That's statistical learning just baked into me that songwriters and stuff play with by tweaking and having little surprising bits in there. So even if the song is something I've heard before, I still want to listen to it again. I still feel surprised. And so is that the same surprise that you get from the surprise of a word that carries meaning? Can't be. So I think that's a real interesting question. It's something that we're thinking about and trying to quantify and parameterize, but we're behind on relative to the speech stuff so far. Yep.
There was another question here.
Yeah, thank you for this very nice talk. So
Wait. Otherwise, Lucia cannot hear.
Yeah. I just wanted to go back to the previous question. So I was wondering whether, this regularity in context, so whether for you it was just a tool, basically, to better capture the response, or whether you think it also has an impact on the brain response you will observe. And so more specifically to Ed, I was wondering whether the TRF response would be robust to a different type of context in speech. So for instance, whether you would observe the same TRF response in more conversational speech, or even when you listen to isolated words. Would you expect the same response, basically? Would it be an ERP, or would you expect it to be contextual? Yeah, basically.
Well, that's a cool question. I do think all of this stuff is probably going on, a bit like Lucia's nice slide there at the end as well, with the different representations potentially in different brain areas, and this stuff that can maybe happen in parallel. So I think some of that's going on potentially in parallel. And again, you can try to model all that by looking at, say, just individual word frequency as a standalone measure of, and so if you just fire words at people, you would expect that to map nicely onto the brain responses. Now, in the context of a story where a typically unusual word is now showing up regularly, a listener will adjust to that and it's no longer unexpected. So I think you can have effects happening at the individual word level and very strong effects at the context level where, a word like bat or something might be absolutely the next word to happen, and then in one context and not at all in the other. And the brain response there will be enormously different. So I think, am I answering the question? Is that what you were?
I was more thinking about, for instance, the temporal structure of speech, because here, like compared to read speech, conversational speech has more pauses and and breaks. And so would you expect that to have an influence on the TRF? Or?
Yeah, it's a great question. So I actually pitched a grant idea about that I didn't get, so I obviously don't have a great answer for you. But because conversational speech, if you transcribe it, it's word salad a lot of the time. There's ums and ehs and then body language, and people will mumble over things, but the other person knows what they're saying and they can indicate that through their body language. And so, strictly analyzing that then in the context of, I think the word is anaphora, is it? It's a word where, I'm not even sure what anaphora, I shouldn't have said it, but the reference, and the he and she, and the person knows who you mean by she, so the word on its own, she, actually carries a lot of meaning because it's reference to something earlier in the conversation. I think you can do it. I think it's a rich area to dive into, and that's what I wanted the money for, but they weren't convinced. Maybe in the future.
More questions, comments from the audience? Ta-da! Yes.
Hi. Regarding these temporal techniques, do you think it's possible to identify fluctuations in attention in this way? So seeing this temporal response function changing over the course of these long recordings with different speech?
Yeah, I think so. So it depends. Our algorithm, our original one was just based on these reconstructions of envelopes for each trial, and each trial was a minute long. And so if you started to try and shrink that down and to try to base it on reconstructions that are 5 seconds long, now you're just dealing with a signal to noise issue. So everything is just a bit worse performing. So lots of labs actually now are trying to do better than and several have done much better than we're doing in terms of decoding accurately with short segments of data, using lots of fancy algorithms that incorporate nonlinearities, incorporate a history term. Who were you paying attention to? And then see if that changes over time to indicate that they might be switching, using other topographic signals on your head to see if you're switching where you're paying attention over to another location, probably then to another speaker. So there's lots of ways to capture other signal and use fancier, shorter algorithms that can indicate attention on shorter time windows. And once you have those shorter time windows, then you can get a sense of who someone's paying attention to much closer to real time, and whether they're paying attention at all maybe as well. I mean, even our semantic stuff tells whether you're paying attention. If you're not paying attention and you're hearing the speech, you've got no understanding signal popping up there. I mean, this is, in part, there's a bit, industry are interested in this too. There's a real push to build hearing devices, either hearing aids or even consumer devices with a few EEG electrodes in them that can have some algorithm on the device that picks up what you're trying to listen to or what you care about or what you're paying attention to improve the processing of, especially for hearing aids, say, because at the moment they largely just boost up everything, and an old person in a restaurant often will just turn it off because they're being overwhelmed. And if you can have an algorithm built on there that's informed by the EEG, then you can start to steer people the signal they're trying to pay attention to, for example. So there's some money going into it, and people trying to decode attention faster. Yeah.
More questions from the audience?
I have one spicy topic I'll throw in for fun. Just I've gotten very I think I've gone too far. The postdocs in my own team correct me all the time, but I've gotten very focused on these temporal changes and the ERP type responses that we're seeing. And Lucia had the nice plot with the 4-word, 5-word, 6-word sentences where you can see this thing peaking. So I have a little pet kind of peeve, I suppose, about people interpreting some of these EEG temporal patterns in terms of the entrainment of ongoing oscillations. And now I know lots of people have written about it, but this loaded word entrainment suggests that there's an oscillation going there already that now is being conducted like a conductor into some peak and trough kind of dance with the stimulus that I think is interesting, and there's lots of evidence for that kind of entrainment in very rhythmic paradigms. But I get worried. My own pet thing at the moment is a big concern that is being extrapolated too much into the domain of naturalistic stimuli that are not that temporally predictable, and that how much of this is really just evoked activity from the kind of things that I've tried to be presenting here versus the entrainment of endogenous oscillations that were already there. I think a lot of people drink that idea up quite quickly and are not as healthily skeptical about it as they might be. So that's my 2 cents on that, if anyone has a reaction.
I can comment on that. I agree with you, but I was discussing this point with the physicist who makes expert in modeling. And so my idea is, in the community, the idea is just to study coupling of simple oscillators. Okay, so you have this idea, you have 5 hertz, and then that you should find this syllabic rate in speech, but while speech is not regular, as you say, so it shouldn't work. And then he was saying, well, but there are different types of oscillators that actually can adapt in time and can change actually their frequency. So in my view as a psychologist, so I'm completely ignorant. I'm a musician, so I thought really about a metronome. For me, oscillator is just a metronome, and say, yeah, simple oscillator can be like this, but in more complex system, you can have also more complex dynamics. So that may also, at least in theory, it's possible that still this similar mechanism of entrainment would be in place. But I completely agree with you that that's an overstatement in general in the community. Maybe Lucia wants to comment on that.
I mean, I'm not, I think I'm just going to reiterate what you guys are saying. And that's what I was trying to present in my studies. At least the way in which I think about it is integration towards a bound, and I think that there, you can think of an ERP tracking this, and it doesn't necessarily have to be an entrainment of an endogenous oscillation. I have the same suspicion that it might actually be difficult to think about that because language and units have such a wide range. So it would have to be, I'm not saying that oscillations don't play a role. Perhaps they do play a role in a narrower, in narrower domains, such as, for instance, syllable parsing, and so on, because that the variance is much smaller. For language as such, it just seems a little bit more complicated. Like it would have to be like really, 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 flexible. Now, but I do take your point that, for instance, in the world of, even if you look at, the very old work of Wolfsinger, when they look at the gamma oscillations, one of the things that they are looking at now is, or thinking of an oscillation just as a excitation inhibition cycle, right? And if you then go with that definition of an oscillation, then, because that to me, comes into the integration window. Excitation inhibition, that's the cycle, right? But that seems a different definition of an oscillation, I would say, right?
I think it's good for the young generation to stop here and to get a flavor of these two big schools, to get that. So there's no oscillations or there's only oscillations. There's only two views about seeing things, And so you have the two communities. So there's no oscillation, guys, or some other only oscillation. Of course, in the middle, there is probably science, yeah? So, and for now, I think we can stop here, and I see Anne-Sophie coming. She has a urgent announcement, or no? No?
Well, she would like to thank again the speakers for high quality presentation. Thank you very much. Thank you for our co-chair for animating the discussion so well. And we have small gifts for you before you leave. We have gifts for all speakers, just in case the hands-on speakers, if they leave earlier yesterday, please stop by the staff booth. We have presents for you. We have a special CuttingEEG2021 And if you guys want to have the CuttingEEG2021 please stop by the booth, staff booth.
You're the one who needs the gift for organizing this, not us.
Okay, give it back. You say everything?
Yes, I still have to say something about the forum. Please note that you can continue the discussions on the forum. This has been created for that as well. And if you did not receive an invitation, please check your spam folders before you get in touch with us. Mm-hmm. And I think...
Yeah, I'm just going to talk about the food. Now it's the lunch. We're 15, a little more minutes late, and we're going to try to start on time, providing the 100% online session of this afternoon goes well. And yeah, so be back on whatever is written on the program. And what?
I forgot to say something.
Well, you want to say it now? Go ahead.
Please.
All right, but I'm not done.
Thank you. I just wanted to say for the online speakers, so please be sure I'm going to try to save some bottles. And you're very, very much welcome to stop by the lab when you come over in Aix-en-Provence. But
do it quick.
Yeah, be quick.
We're going to drink them all. Yeah, and also what I wanted to add is that be sure to stop by the posters. And I think today it's all of you have odd numbers, like 1, 3, 5, etc. You're supposed to be, it's for myself just to be sure. So yeah, you're supposed to be around your poster today, and the even numbers, 2, 4, you're supposed to be there tomorrow, okay? Don't eat all the food, the first that goes to the buffet. We increased the, it seems that you like it a lot, so we increased the number of portions, but don't take 5 just for you. All right? All right, have fun. See you later.